Hello students, welcome to our online video tutorial class. I think you have already seen the previous videos on the chapter Sexual Reproduction in Flowering Plant. You can remember in the previous video we have discussed about the structure of ovary. We have also discussed about the structure of pollen sac, pollen grain. We have discussed about the pollen viability, as well as pollen sac structure, how the pollen grains are forming within the pollen sac. Then we have discussed also about structure of pistil and various types of flower based on the number of ovary chambers, and all these things we have discussed in the previous video. So I think you have studied that video well. So today I have decided that I will be just starting from the next part, that is the structure of ovule. So first of all, let's see the structure of the ovule. You can see here the ovule, that is the specific structure which is commonly present within the ovary. That ovule in future is producing the seed. Now the point is, this ovule, how it is looking, or whatever the structural features are present in the ovule. First of all, we have to keep it in our mind that ovule is covered by the skin-like layer which is commonly known as integument. First of all, this integument can be single layered. On that time the ovule is commonly known as unitegmic ovule, and if the integument are two layered, that means double layered, on that time it is commonly known as bitegmic ovule. So based on the number of integument, we can separate or we can segregate the ovule into two different categories, unitegmic and bitegmic. Coming to the next point. Next point is about nucellus. What is that? Nucellus is actually the protective covering surrounding the embryo sac of the ovule. Suppose this is the embryo sac. So surrounding the embryo sac, this specific layer of cells which are actually present, which are actually protecting the embryo sac, that is commonly known as nucellus. Next, inside the nucellus, centrally located embryo sac is present. So embryo sac is there, which is present in the central position of the ovule. Ovule is held by a specific type of stalk-like structure that is commonly known as funicle. But here one point we have to keep it in our mind, that if the ovule is anatropous ovule, anatropous ovule means if the aperture is present towards downward direction, on that time that ovule is commonly known as anatropous ovule. Again I am repeating, if the pore, this specific pore whatever I am showing, that is commonly known as micropyle. If this specific micropyle pore is present or facing towards downward, on that time it is commonly known as anatropous ovule. So in case of anatropous ovule, this specific stalk is just creating a bend here or bending here, and then it is attaching with the ovule. So this specific bent portion of the stalk, that is funicle, is known as raphe. So what is raphe? Raphe is the bent portion of the funicle which is attached with the anatropous ovule, is commonly known as raphe. Going for the next point, that is micropyle. We know this is the aperture through which the pollen tube is commonly entering within the embryo sac. It is commonly entering within embryo sac through this aperture, and micropyle is present in which end of the ovule? That end is commonly known as micropylar end. So micropyle, you can see in the diagram, it is present here. So this end will be considered as micropylar end, and only the opposite one, this specific opposite region is commonly known as chalaza, which is present opposite to the micropyle. This specific pole is also known as chalazal pole or chalazal end. So this is the overall structure of the ovule, where we can see ovule can be surrounded by a single layer integument or double layer integument. Second, there are multiple cell layers surrounding the embryo sac, and that is commonly known as nucellus. Next, embryo sac is present centrally to the ovule. Ovule can be held by a specific stalk that is known as funicle or funicle. In case of anatropous ovule specifically, this specific funicle is bent and getting attached with the ovule, and this specific bent portion is known as raphe. Next, micropyle is the aperture through which the pollen tube is commonly entering within the embryo sac, and this aperture is present only for that purpose, and in which end that micropyle is present, that is known as micropylar end, and only the opposite one. Okay, so that specific opposite end is commonly known as chalazal end or chalazal pole. So this is the overall structure of the ovule. Going for the next slide. In the next slide we will be discussing about the structure of embryo sac. So let's see. This is the embryo sac structure which is commonly present within the ovule. So let's see whatever the structural components we can see within the embryo sac. First, it is obviously present within the ovule, we all know, containing some nuclei. Actually, if you start counting, total there are 8 nuclei and number of cells are 7. How? 1, 2, 3, 4, 5, 6, 7, 8. These are the 8 nuclei and number of cells. These 3 cells total 6, and this one is total 1. That means middle 2 nuclei are present within one cell. So total 7 cells. So that means embryo sac structure is composed of 7 cells and 8 nuclei. Now, first structure is egg. This egg is actually the structure which is producing zygote in future, that is present towards the micropylar end. You can see here this yellow color cell, that is the egg. In future it will be forming only the zygote and it is present towards the micropylar end, because through the micropyle the pollen tube when enters, within the pollen tubes male gametes are present. So the male gametes will be fertilizing with this egg to form the zygote. Next, synergids. Synergids are these 2 orange colored cells which are present on the both side of the egg cell, and these 2 synergids are exclusively present for providing nourishment to the egg cell. Here, one point we have to keep it in our mind, that these 2 synergids are containing one specific finger-like projection in their base, that is known as filiform apparatus, and that filiform apparatus is actually responsible for providing the nourishment to the egg. So these 2 synergids are only for providing the nourishment to the egg, they are present. Next, centrally located 2 nuclei, those are known as polar nuclei, and the entire cell is known as polar nuclei cell. These 2 polar nuclei, what is their function? Their function is exclusively producing the endosperm, because when the 2 male gametes are entering within the embryo sac, first male gamete is fertilizing with the egg cell, second male gamete is fertilizing with these 2 polar nuclei and forming triploid endosperm. So for producing endosperm only, these polar nuclei are present. Next, presence of antipodal cells. These green colored cells what are present towards the chalazal end, those are commonly known as antipodal cells. Till yet proper function of the antipodal cell is not properly understood or not properly discovered. These 3 cells are only present, they are only seeing the entire mechanism of double fertilization which is happening within the embryo sac. So antipodal cells are these 3 who are only viewer, you can consider as a viewer of the double fertilization process. So this is the entire embryo sac structure containing 8 nuclei and 7 cells. I think the entire structure of the embryo sac from the figure as well as what is written here in this slide, you have understood all. Next, we are going for the next slide, that is megasporogenesis or formation of megaspore. So let's see how this megaspore, that means embryo sac, that is forming. The process of embryo sac forming, that is commonly known as megasporogenesis. So let's see. Here we can see, first of all, I have written ovule. Keep it in your mind, this ovule is actually, within the ovule there is a cell which is present towards the micropyle. Those are initially called as archisporial cells. So within the ovule there are few cells which are present towards the micropylar end before presence of the egg and synergid. Those cells are commonly known as archisporial cells. These are the primitive type of cell. This archisporial cell is dividing into 2 different type of cells. One is primary parietal cell, another one is primary sporogenous cell. Now this primary sporogenous cell, you can understand from the name, that means it is generating some specific structure, that's why sporogenous. So what is actually generated by it? It is generating the megaspore mother cell. When we have discussed about the pollen grain formation, we have studied their microspore mother cell, because microspore mother cell is forming the microspore or pollen grain. Here megaspore mother cell is the type of mother cell which is exclusively responsible for producing the megaspore or the embryo sac. Now the point is, this megaspore mother cell is forming megaspore tetrad. Obviously by the meiosis cell division, meiosis 1 and meiosis 2, these 2 cell divisions occurred sequentially, and after that megaspore tetrad has been formed. Here also this specific megaspore tetrad which has been formed, that is not present in the tetrahedral arrangement like the microspore tetrad, rather it is present in a linear arrangement. Now the point is, this megaspore tetrad is differentiated into 2 different groups. One group is containing 3 megaspore, another group is containing only 1 megaspore. Now the point is that group which is containing 3 megaspores, there you can see 3 megaspores will get degenerated, they will get destroyed because they remain in the inactive form. But another megaspore which has segregated from the remaining 3, that specific megaspore is getting activated and forming the functional megaspore. That means during the microsporogenesis we have seen that specifically 4 microspore or pollen grains which are actually formed, those all are active. But in case of megasporogenesis, keep it in your mind, megaspores are also produced 4, but those 4 all not remain active, only 1 remain active, remaining 3 are get degenerated because only 1 is required for the process, and for that reason one is remain activated, remaining are getting deactivated. So this is the entire mechanism of megasporogenesis or megaspore formation or embryo sac formation. Now I am just moving for the next slide where we will be discussing about the development of the female gametophyte. That means when the megaspore or embryo sac is formed, inside megaspore there is only one cell with one nucleus. Now from that one cell and one nucleus, how 7 cells and 8 nuclei are produced within the embryo sac, whatever the structure just in the previous slide we have seen, that specific structure how it is forming, let's see. First of all, you can see here I have written the megaspore containing single cell, single nucleus. Keep it in your mind, this megaspore is dividing 3 times. Again I am repeating, megaspore is dividing for 3 times, and that is also by the mitosis cell division. When they are dividing by the mitosis cell division for the 3 times, automatically 8 daughter nuclei is getting formed because 1 to 2, 2 to 4, 4 to 8. Just the 8 daughter nuclei has been formed. From those 8 daughter nuclei, 4 nuclei move towards the chalazal end of the embryo sac and remaining 4 nuclei move towards the micropylar end of the embryo sac. Now, whatever the 4 nuclei move towards the chalazal end, from there only 1 nuclei has come to the central position of the embryo sac, that is known as central nuclei. Another 3 or remaining 3 remain in the chalazal end. Same in case of those nuclei which are present in the micropylar end, from there also 1 nuclei has come to the central position, remaining 3 present or remain within the micropylar end. Now the point is, whatever the 3 nuclei present in the chalazal end, those are further forming the antipodal cells, whatever you have already seen in the figure of embryo sac, because those are commonly present in the chalazal end. They don't have a specific function, so they are present in the chalazal end. 2 central nuclei which have come from both of the ends, those are making the polar nuclei, and those polar nuclei is getting surrounded by a plasma membrane forming the polar nuclei cell. And the 3 nuclei which are present towards the micropylar end, out of those 3 only 1 is getting converted into egg, that is the middle one, and both of the remaining cells which are present on the both of the sides of egg, those are getting converted into synergy, that is giving the nourishment to the egg. So this is overall the segregation of the nucleus as well as cell which is happening within the structure of embryo sac, and in this specific flow chart we are actually understanding that how the antipodal cell, egg, synergid, polar nuclei, these kind of structures have been developed within the embryo sac. So this is the megaspore development process. Okay, I think this video, this specific slide you have understood properly. Going for the new topic, and another new topic, that is pollination. We all know pollination is the process of transferring the pollen grain from the anther to the stigma of the same flower, or from one flower to another flower of the same plant, or maybe to the different flower of the different plant. So that can be called as pollination. So pollination is the mechanism by which the pollen grain from the anther is getting transferred to the stigma of the same flower, or the different flower of the same plant, or maybe to the different flower of different plant. That is commonly known as pollination. So let's see the next slide what is written. Pollination is actually of 2 types. One is self-pollination, another one is cross-pollination. Self-pollination is subdivided into 2 categories. One is autogamy, another one is geitonogamy. Next, Autogamy is again getting subdivided into two different categories. One is chasmogamy and cleistogamy. Chasmogamy is such kind of pollination where the flower is remain open, and cleistogamy is one kind of flower where the flower remain in closed condition. And cross pollination is commonly known as scientifically or biologically as a xenogamy. About each and every definition I am not saying in this slide, because in the next slide I will be discussing about the definition of each and every kind of pollination. So I think you have understood properly the types of pollination here. Self-pollination, cross-pollination. Self-pollination can be subdivided into autogamy and geitonogamy. Autogamy is subdivided into chasmogamy and cleistogamy. And cross-pollination can be called directly or biologically xenogamy. Here you can see the definition of the self-pollination. Self-pollination means when the pollination is happening between the flowers of the same plant, or rather you can say within the same flower. That means when the pollen is transferred from the anther to the stigma of the same flower, or maybe of the different flower but in same plant, that is called as self-pollination. What is about cross-pollination? Obviously, if the pollen grain from the anther is getting transferred to the stigma of another flower of another plant body, then it is known as cross-pollination, which is commonly known as xenogamy or allogamy. Here is another term you can understand or you can see, that is allogamy. That is also the synonymous term of the xenogamy. Now the point is, this self-pollination can be subdivided into two categories. Already I have seen in the previous slide also, autogamy and geitonogamy. What is autogamy? Same. In case of self-pollination, what I have told that when the pollen is getting transferred from the anther to the stigma of the same flower or the different flower of the same plant. Now just I am breaking the definition of the self-pollination. If the pollen is getting transferred from the anther to the stigma of same flower, then it is known as autogamy. And if the pollen grain is getting transferred from the anther to the stigma of another flower but of the same plant body, then it is known as geitonogamy. So autogamy and geitonogamy, these are the two types of self-pollination. Only the flowers are getting different. In autogamy, within same flower the pollination is happening, and in geitonogamy, in between two different flowers the pollination is happening, but obviously both of the flowers are present in same plant. Okay. And allogamy, we all know, obviously pollination is happening in between two flowers, and these two flowers are present in different plant body. Going for the next slide. Here you can see properly what is self-pollination. You can see here within the same flower the pollen is getting transferred from the anther to the stigma, and in case of cross-pollination you also can see from the anther of one flower the pollen grain is getting transferred to the stigma of another flower, but in the different plant, not in the same plant. Okay. Next we can see here the self and cross pollination types. Here autogamy and geitonogamy is also very much clear. Autogamy means when the pollination is happening within the same flower, then it is autogamy. But when the pollination is happening in between two flowers, you can see here two flowers but in the same plant body, then it is geitonogamy. And cross pollination, already we have seen from one flower to another flower and both of the flowers in different plant body, that is the cross pollination. So only diagrammatically it has been shown here what is cross pollination, what is autogamy, what is geitonogamy. Next we are discussing about only chasmogamy and cleistogamy. What is about them? Chasmogamous flower or chasmogamy is a such kind of flower which always remain open at the mature condition. Their stigma and stamen remain always open. In case of cleistogamous flower, they are bisexual flower, and that's why they remain in the closed condition, because the anther and stigma both are present inside the flower. Here one thing we have to keep it in our mind, that both of the flower is autogamous type, because autogamous flower has been subdivided into chasmogamous and cleistogamous. We have understood that the pollen is getting transferred from the anther to the stigma of the same flower, but if the flower remain opened, if the stigma and stamen both are remain exposed, then it is chasmogamous flower. And if the pollen grain is transferred from the anther to the stigma of the same flower and the petals of the flower remain closed, so that inside the flower that transfer of the pollen grain is happening, then it is known as cleistogamous flower. In case of chasmogamous, it is written they may be cross-pollinated or self-pollinated. Mostly they are the type of self-pollination. Cross-pollination mostly not happen, but when the flower is remain open condition, obviously open flower is supporting self-pollination first of all, as well as cross-pollination. But in case of cleistogamous flower, keep it in your mind, when the flower is present in the closed condition, obviously it is supporting the self-pollination only. It is not supporting cross-pollination. This is the slide where you can see the flower structure. This is the chasmogamous flower where the petals are opened, and this is the cleistogamous flower which is found in case of viola. And here you can see all of the flowers remain closed. They are present in the bud condition, so that the anther and stigma all are present inside, and the self-pollination can only happen. Cross-pollination can't take place. So this is the difference in between chasmogamous and cleistogamous flower structure. Going for next slide, that is the agents of pollination. That means here we will be discussing about whatever the various agents are actually responsible for this pollination process. So let's see. First of all, we have to know there are two types of factors are commonly present for the pollination. One is abiotic factor, another one is biotic factor. Abiotic factor means those are the non-living factors. Okay, they don't have any life, and those specific factors, as an example wind, as an example water, those are also causing pollination, or those are also helping pollination. So if wind is helping in the pollination process, on that time it is known as anemophilous flower, and the process is known as anemophily. And if water is helping in the pollination process, on that time that process is known as hydrophily, and those flowers are commonly known as hydrophilous flower. Coming to those factors which have life, and those specific factors are responsible for cross-pollination. Exclusively these are the agents of pollination. And which kind of pollination? Of cross-pollination, because for self-pollination no agent is needed. So first one you can see entomophily, that means the pollination when it is happening by the insects. And keep it in your mind, those flowers which are pollinated, cross-pollinated by the insects, those are known as entomophilous flower. Next you can see ornithophily, that means if the pollination is happening by the help of birds. And keep it in your mind, this kind of flower which are pollinated by the blood, those are known as ornithophilous flower. Cheiropterophily, that means this is such kind of pollination where bat is actually helping in the pollination process, and those flowers which are get pollinated by this process is known as Cheiropterophilous flower. Next, malacophily, this is another pollination type where the malacos means mollusk organism, snail and slugs, they are actually helping in the cross-pollination process, and those flowers which are commonly pollinated by this malacophily process, those are known as malacophilous flower. And the last one you can see myrmecophily. Myrmecophily is a such kind of pollination where the cross-pollination is happening by the ants, and those flowers which are pollinated by these ants, those are known as myrmecophilous flower. So now the point is, here we will be discussing about each kind of pollination except myrmecophily, because that is not there in our syllabus. So let's see the next slides. First you can see here the anemophilous flower. See the respective features of the anemophilous flower which are pollinated by the wind. Anemophilous flowers don't have the color to attract the insects because they are not get pollinated by the insects. They don't have scent or nectar because these things are not needed because they are not attracting insect. They have numerous small flowers. Small flowers are enough for them. Stamens have long filament and are exposed. Stamens are containing long filament and they are exposed. Why? Because if the filaments are not coming to the touch of wind, obviously wind can't help in their pollination process. For that reason, their pollen grains are dry, lightweight and powdery. Obviously their pollen grain is dry, lightweight and powdery because of for their lightweight they can flow in the wind. Okay, they can flow with the air and get transferred to another flower of the another plant body. So for that reason these features are commonly present in them. Stigma is sticky and feathery. Stigma is sticky as well as it is containing feather like structure so that it can glide in the air. It can move through the flow of air or wind. So these are the basic features whatever we have to mug up for anemophilous flower. I think every feature we have understood properly. So let's see the next slide. In this slide we can see these are the feathery stigma. The stigma is actually feather like structure containing. Why? So that the pollen grains which are actually coming from the surface of the anther, so those specific pollen grains are getting transferred through the air. As well as why the feathery stigma? Because of due to presence of this feather like structure, what is happening when the pollen grains are just moving in the air, accidentally only they will be getting associated with the surface of these feathers which are present actually on the surface of ovary. So due to presence of this feathery stigma, obviously they can capture the pollen grains from the environment directly, and these are the anthers. From this specific anther, these are commonly known as versatile anther. Obviously these versatile anthers are responsible for the pollen grains which are actually coming out. Okay, and those are all helps in those are actually moving in a gliding process, and those specific type of pollen grains are in very lightweight, they are dry and powdery appearance they have. Example, in case of monocot grass, in case of maize, in case of jowar, that means in case of most of the monocot plants like grass, maize, jowar, specifically this kind of anemophilous pollination is taking place, or anemophily is taking place. Moving for hydrophilous flower, this is another type of flower where the pollination is happening by the water. Here the flowers are colorless because obviously when it is pollinated by the water, no color is needed. They are small in size and they are present without nectar and fragrance. Next, pollen grains are long ribbon like structure, that also we have seen in the previous slide, which are carried with the current of water. So pollen grains are very much long ribbon like and the very lightweight containing, so that they can be carried by the water current very easily. The pollen grains are light but covered with waxy layer, so that it can be protected from the wetting. That means when it will be covered by waxy layer, obviously it is that pollen grains are getting waterproof, and due to this waterproof appearance, obviously it will not be getting destroyed by the water. So this is the basic features for the hydrophilous flower. Going for the next, here you can see one aquatic plant is given, that is example of Vallisneria or hydrilla. So Vallisneria, hydrilla, potamogaton, myriophyllum and water lily. Again I am repeating, hydrilla, Vallisneria, potamogaton, myriophyllum and water lily. These are the examples of hydrophilous flower which are commonly pollinated by the water. So I think the anemophilous and hydrophilous flower you have understood very clearly. Whatever their basic features should be present, that also you have understood. Okay, so I am just recapitulating whatever the points we have studied today. We have studied about the structure of ovule, we have studied about the structure of embryo sac, we have discussed how the megaspore can be formed, that is embryo sac can be formed. We have discussed about female gametophyte development, that means how the nuclei and cellular structure are coming within the embryo sac. We have discussed about the pollination definition as well as its types, and each and every type definition also we have discussed in this following slides. Next we have discussed about the type of agents involved in the pollination process, and out of that only abiotic factors which are the responsible for pollination process, those I have discussed in this video. Next video I will be starting from the pollination again, and I will be discussing at first the various biotic factors or biological factors which are responsible for the pollination, and then I will be moving so forth. So I think the entire video whatever I have made you have understood properly. So please study about this video, and you have enjoyed this video a lot I think. So thank you everyone.